Hello, and welcome to this little micro lab, if you will. My name is Brendan Ganton, and I am an instructor with the School of Environmental Sciences at Lakeland College here in Vermilion. We're putting together a series of videos to kind of give prospective students or our current students not only a good laugh, but also expose you to some of the techniques, some of the things you might be learning in our labs as part of our awesome hands-on experience, hands-on learning that we give you guys at Lakeland College. Um, so here we go. Today we're going to be talking about salmonid ID, salmonid identification. What does that mean? We're actually talking about a subset of the fish here in Alberta that we would call or typically refer to as trout. You guys are used to probably referring to all these fish as trout, but really we're talking about the true trouts and the char species here in Alberta. So it's great as anglers, also as fisheries technicians, environmental technicians. Important for us to be able to reliably and easily distinguish between different species of fish here in Alberta. And so one of the things that we'll go over in our fisheries labs is our trout, our salmonid ID. So I titled this one, Which Fish is Which? I like to start with a story. And so this is a story of a young fish biologist, truly, not even a fish biologist yet. And yes, it's right. That young fish biologist is actually myself. Very early on in my fisheries career, I would have just graduated from the University of Alberta. I transferred to the U of A from Lakeland College. Great degree transfer program. Took all of my courses from here, went there, and finished. So I was looking for a fisheries job. I had worked already in northeastern Alberta, so I was really familiar with our warm water fishes. But because some of my classes had been, 2, 3, 4 years previous, and I wasn't necessarily the best student, if you can imagine that. Um, I was actually fairly weak when it came to my salmonid identification, our species of the eastern slopes. And the job I was applying for did require me to reliably, consistently identify those fish from photographs, sometimes black and white photographs. So it was a very important skill for this job. As part of the interview, I sat down, and at one point they said, "Okay, sit down. We're going to give you a fish ID test." And so thinking back, I was like, "Okay, fish ID, I can do this. I remember there were a lot of key distinguishing features. And here's the first fish that I might have looked at on that test. It may have been this very photo." And so I started thinking through that process, key distinguishing features. Oh, this fish has red dots on it. I know there's something about red dots and it means something. Couldn't quite remember what. Par marks, those dark bars along the side. I was like, par marks, that coloration means something. White fin edges. I knew for sure. White fin edges. Yeah, that means that it's probably, I think it means it's a char. Blue halos. There's blue halos on those red dots. What that meant something. Black wormy things on the back and on the dorsal fin. Red dots, back to the red dots. Like, what did they mean? I couldn't quite remember. Yellow dots, double rainbow. What does it all mean? I was completely overwhelmed, stressed out, couldn't remember really anything. My brain kind of shut off. And at the end of the day, hypothetically, this is just an example, but I didn't do very well. Let's say I got 13 out of 20. That probably was better than I did. But I didn't do very well. And everything else in the interview went really well. They felt like I would be a good fit for the job, except I really couldn't identify my fish very reliably. And luckily for me, they didn't just throw me out. They had some really experienced, great biologists in Rocky Mountain House and just south of there, who had just finished working on putting together basically a new method or a new approach to consistently identifying the trout species of the East Slopes. And they put this together for anglers to help determine which fish need to be released from those that we could harvest or those that we might want to remove from a system in this case. What went wrong in my test? I had overwhelmed myself by trying to remember every single detail about the fish. when there may have been an easier approach. And that's what the biologists in Rocky Mountain House introduced me to, and the same thing that I take our students through. So our objectives for our mini lab today is to contrast and compare key features of trout and char in Alberta. We want to apply a consistent and logical approach to identifying salmonids. And above all, we want to remain calm under pressure. So here is effectively the simple system that the biologists introduced me to. And really, it was about creating a system. So I like to say to my students, don't memorize the individual features for each trout and then try and pick out exactly which one it was. Let's start with a system. So don't memorize, systematize. Systematize. I think I made that word up. It's not a real word, but it is for today. Systematize, because it works. Don't memorize, systematize. So what we see here is basically this partial key for the trout and char here in Alberta. There's a few notable exceptions that I skipped over, like our whitefishes, and also some of our newer introduced species like tiger trout or golden trout aren't on this key. So this is a partial key, but it definitely hits on our most common species that we're going to come across. Partial key for trout and char. And it breaks it down into an easy system, a series of decisions. And the first one being black spots on the sides or no blacks on the spots on the sides. If there are black spots, we know it's a true trout. If there are no black spots on the side, we know it's in the char, okay, the char side. So all those trout that we're used to calling trout, bull trout, lake trout, brook trout, they might be better referred to as bull char, lake char, and brook char, but common names are a weird, fickle thing. So bull trout, lake trout, and brook trout they are. How do we use this system? Again, those easy, simple things. Step one, black spots, no black spots. If it has black spots, then we take a look at the next level. Does it have pale halos around those black spots? If it does, we're already done, and we know it's a brown trout. If there are no pale halos, we have one more question to answer on the trout side of things. Does it have a red-orange slash under its jaw or no red-orange slash under the jaw? If there is that red-orange slash, it's a cutthroat trout. Red slash, cutthroat trout. Easy to remember. No red-orange slash under the jaw. Rainbow trout. Let's jump back over to the char side. So we've decided if there are no black spots, step one, it's a char. Then we have the next question, question number two. No spots or markings on the dorsal fin, or there are spots or markings on the dorsal fin. The dorsal fin just being just that. There's a great picture of it there. That central fin on the center of our salmonid's back. No spots or markings, that's a bull trout. That's where that saying no black, put it back comes from, because there's no black spots on the sides and there's no spots or markings on the dorsal fin. It's a bull trout. What about the other side? If there are spots or markings on the dorsal fin, that leads us to one more question on the char side. So question one, question two, question three. Are there pale spots on the dorsal fin and the tail, with the tail being deeply forked? If so, that's a lake trout, right? That's a giveaway. That lake trout tail, if you've ever fished for them, that's a real nice giveaway for our lake trout. But what if there are black markings on the dorsal fin? They're not as pale. They're actually black markings on the dorsal fin, and our tail's not deeply forked, more square in shape. And you can take a look down on the bottom right. Yeah, that's a brook trout. So no black spots on the sides. If we have markings on the fin and they're black markings with a not so deeply forked tail, that's a brook trout. That's really the ease of the system. Those few questions can lead us pretty consistently to identify our fish correctly. Let's run through this a few times. Look at this little guy. What kind of species is that? Young fish technician Brendan panicked. I actually probably got this one right, okay, just because we see it so commonly. But let's put it up on the system. Black spots on the sides. Yeah, it's on the trout side. Okay, do we see pale halos around those black spots? We don't in this case. So no pale halos. Then we're looking at that under the jaw, red-orange slash. And in this case, there isn't one. Yeah, it makes sense. That looks like the fish that we were pulling out of the ponds as we were growing up, a rainbow trout. We do have native rainbow trout here in Alberta, the Athabasca rainbow trout, but the vast majority, and they really only exist in the upper Athabasca River drainage. Most of our trout that you're going to catch in ponds have been introduced, rainbow trout. Let's take a look at another one. We have two fish here. And think back to me with the red spots and the blue halos and all of that stuff going on. These two fish superficially might look very similar. And this is a real sticking point or a hang up for young fisheries technicians. But let's see, what do we have here? Black spots, that first top left fish, black spots on the side, so we know it's on the trout side. Pale halos, yeah, we've got pale halos around those black spots. We're looking at a brown trout. Bottom picture there, no black spots on the sides. Oh, we know it's on the char side. Spots or markings on the dorsal fin. Yeah, we have them, those black worm-like vermiculations on the dorsal fin. And if we take a look under that water, they are black markings, and the tail's not so deeply forked. So we're looking at a brook trout. Okay. What about this fish? One of the easiest ones now that we're used to the system, because there's no black spots on the side. Then we look at that dorsal fin. Hey, and there's no spots or markings on that dorsal fin. No black, put it back. That's a bull trout. That's our provincial fish of Alberta. Easy to identify him now. So a quick recap. Black spots on the side of the body, that first decision that we made, tells us these are the true trout, cutthroat trout, brown trout, or rainbow trout. If we're lacking those black spots on the sides, we're talking about a char, which we commonly refer to still as trout, the brook trout, the lake trout, or the bull trout here in Alberta. Show me what you know. I've got, I think, about 3 fish here. So who am I? Which type of fish is this salmonid? Let's see what we've learned. Give you a couple seconds, and then I'll start putting up some little hints. Think back to that system. No black spots on the side of his body. There are some spots or markings on this dorsal fin, and they're pale, and it does have that deeply forked tail. If you can't quite remember, there's a cheat sheet just for you guys. But yeah, we can tell that is a lake trout. What about this fish? Who am I? Think back to that system. Black spots on the side. Then pale halos. We have both those things, so we're already done, right? You remember? It's a brown trout, even though it's decidedly not very brown, this one. How about this fish? Black spots on the side? We've got that question down. We remember it now. There are black spots on the sides of the body, but there's no pale halos. That left us one more question. Do you remember what it was? Yeah, red-orange slash under the jaw, so we know it is a cutthroat trout. Thanks for showing me what you know, and thanks for joining me for this small micro laboratory, just highlighting some of the things you might learn as a wildlife and fisheries conservation diploma student here in environmental sciences at Lakeland College. So just to recap, here's what I took you through. We wanted to contrast and compare those key features of trout and char in Alberta. Then we applied a consistent and logical approach to identifying salmonids. Remember that word I made up? Don't memorize, systematize. Lastly, we remain calm and cool under pressure. Thanks so much for joining me. My name is Brendan Ganton. You can get a hold of me, send me any questions you might have regarding our presentation today, or any questions about becoming a student at Lakeland College. Thanks so much. Bye-bye.